Hi, the following content is for educational purposes only and does not constitute medical advice. If you are a healthcare provider, please consult with your affiliated institution or local ethics committee for ethical guidance related to clinical practice.
Hey, BioE 131, this is Russ, and I'm here for technical briefing module number 2, which is on newborn screening. Just a few things to talk about before we get into the ethics. What are we talking about here? Why newborn screening? There are inborn diseases. That is to say, the baby is born with a disease at birth that can be very severe, but is treatable. In order to capture those, you have to make the diagnosis very quickly and then make an intervention. So what do? Well, right now, everybody who's born in California and really across the United States gets what they call a heel stick, which is literally a puncture in the little tiny foot. There's the little tiny foot of the baby. They put a needle and get a little bit of blood. I guess I should use red here for the blood. A little bit of blood, and then they put this on a card. Several of the spots, they're red. And then they can send these to labs that then will do tests for various compounds that if they are too prevalent in the blood, if there's too much or not enough in the blood, they know that the baby is having a problem. So for example, there's a carnitine, there's a enzyme called carnitine transferase, and there's a deficiency of that. And if you see a deficiency of that, then the ratio of free to bound carnitine is a small molecule in the cell, will be abnormal. And that's often checked with mass spectrometry. Okay, and there are tens or hundreds of these chemicals that are checked in the heel stick with these different experimental technologies as a screening test. And that's very important. I'll talk about that in a second. As a screen to see if the baby needs to get further workup. If that screen comes back positive, then they will do diagnostic tests which confirm the diagnosis. So a screening test, just to make this point, is one that is very sensitive, high sensitivity, which means there is a pretty low false negative rate. What is a false negative rate? If somebody has it and the test says they're okay, that would be a negative. That is false because they really have it, but the test said, no, this is very prevalent right now with COVID because people are worried that there are false negative COVID tests where we tell somebody, you're fine, you're fine, you do not have the disease, negative test for the disease, and that's a mistake. That can also happen in screening. You don't want to screen a baby and say the baby looks fine. However, you would tolerate false positives, positives that are false, because there, the diagnostic test, which is usually more expensive and more accurate, would give you the final answer. Okay, so screening tests, low false negative, they tolerate false positive, diagnostic accurate, so they should have very low false negatives, and actually they should have very low false positives too. They should just be on the nail. So what's the new idea for newborn screening? The new idea is instead of taking blood and doing these chemical tests, as I said, there's only hundreds of those available. Why don't we screen by whole genome sequencing? Okay, so the idea there is that these are all genetic defects for the most part. And so if we can get a rapid sequence, this is like within 24 hours, get a high quality sequence, then we could just look at all the genes, look at the sequence of the key genes for mutations that are going to cause trouble. Now, let me just say a little bit about the technology of sequencing technology. The most common is what we call short read sequencing. All of this you can Google, so I'm going to give a very short read sequencing is the genome is chopped up into length about 150 base pairs. And the way you get the full sequence is by tiling these 150 base pair sequences over the entire 3 billion bases. Remember, a human genome has 3 billion bases. And so that's a lot of 150 base pair fragments. And then with these overlaps, you can come up with the sequence of the patient, or of the baby in this case. The problem there is that these are very accurate, but if there are repeats that are longer than about 150, that is to say, there are elements of the genome that are identical. So this is A and this is A2. And if A is much greater than 150, it's going to be hard for you to know the number of repeats. And I'll let you think about that and talk to your study group if that's not immediately obvious. But long repeats are a problem for short read. The emerging thing now is long read sequencing. This helps a lot because these are much longer fragments, and therefore they will not, whoops, whoa, look at that. They will not have this problem because now A can be much bigger before it's a problem. The problem with long read sequencing is it has an increased error rate. It's currently more expensive, and it's not as high throughput yet. But people are working on ways to solve that. Okay, so the idea, just to go back to our idea, is to screen by whole genome sequencing. Let me highlight that to remind you what we're talking about. Oh, that was supposed to be red. There you go. Screen by whole genome sequencing. All right, so what are the challenges to screening, to whole genome sequencing for newborn screening? Challenges. First challenges are what happens if you see new mutations in a key gene of unknown significance. So you're going to sequence, you'll find a mutation. If it's a known mutation, that's fine. You know the clinical implications of that. If it's a new mutation, you don't know what to do. And you have to make a decision about whether this is likely to cause disease or not, and you're going to have to go to the diagnostic test, which is fine because that's what you would be doing anyway. So that's one issue. The other issue is what happens if there are multiple genes that are interacting to create the disease. We might not know how to combine information about all of their mutations. Multiple genes may be multiple mutations, and therefore not sufficient understanding of biology to know whether this is a positive test or a negative test, and therefore you might be nervous and you might go to a diagnostic test and you might do many more diagnostic tests because you're nervous, and that could cost a lot of money. So the screening test can wind up killing you if you wind up doing too many diagnostic tests. Of course, another problem would be sequencing mistakes. You just make errors that make somebody look like they have a mutation when they don't. And the other thing, which is very insidious, is since you're looking at the whole genome, you're going to see mutations all over. And the question is, what is your duty to act on these mutations that are all over other places of the genome? And especially if they have unknown significance. But you see them, and do you report them? Do you ignore them? That's ethics. I'm not going to be talking about that. So that's a summary of newborn screening, why we do it, how we do it with the heel stick, Currently, we're doing lab tests. There's a lot of idea about moving this to sequencing, but there are unsolved questions, and that's going to be a main part of David's lecture in this area. That, BioE 131, is Technical Briefing Module 2. Thanks very much. I'm now going to stop.